All right, welcome back to Chapter 1, Part 2. So this time we're going to talk about some fairly complicated stuff. A lot of it is going to review what we talked about in Structure 1 or 2. And then if you've had ex-phys with Dr. Kephart, a lot of this is going to be really familiar as well. If you've not, or if you just don't remember the stuff that you talked about in ex-phys or that we talked about in Structure, I'm going to try to make it as simple and straightforward as possible, but some of these concepts are a little complicated. So this first slide, though, talks about a lot of the things that we discussed last time. The concept that if you apply an exercise stimulus, so probably the easiest way to understand this is this particular part of the concept is if you go lift weights. Let's say you do 3 sets of 10 of bicep curls or something like that, pretty straightforward one joint exercise. At the end of that, in the several hours after, you're going to see that you have a reduction of strength in the biceps muscle, right? So reduction of strength, reduction of power. And so what we've got there then is the stimulus was our 3 sets of 10 on curls, and then we're going to fatigue, so we get a decline in performance. But over the course of the next several days, we're going to repair the damage. So if there was any structural damage to the muscle cell, we're going to rebuild it. Potentially, we're going to start working on adding some new contractile proteins, more actin, more myosin, laying down some more collagen, those kinds of things. And so then that is compensation in response to that stimulus. So we damage the muscle, now we got to rebuild it. And then shortly thereafter, we're going to get a super compensation. So you're a little bit stronger than you were when you did that initial exercise bout. That's super compensation. And so the idea there is that we have adapted again to that initial stimulus. Probably not fully, right? So as I mentioned last time, you want to change the exercise program at least every 4 weeks, potentially every 2 weeks. But if you're doing your 3 sets of 10 at 65 pounds, you could probably, and you're, let's say, fairly sore on week 1, you could probably do that same weight and sets and reps. So again, 3 sets of 10 for 65 pounds for a second week, and still get some training adaptation, still get a little bit of a training stimulus that fosters adaptations to that load. But beyond that point, you may need to add another set, you may need to add more weight, those kinds of things. But again, in response to that, you're going to get super compensation, you're going to be a little bit stronger. But if we don't apply another stimulus here after super compensation, we get involution. And so involution is just loss of the training adaptations that we made. So as I say in several classes, everything as far as the body is concerned is use it or lose it. Now, eventually you kind of lose everything, right? But the longer, you can hang on to things longer if you continue to use them. So if you go and lift weights for several weeks to months, the muscles are going to adapt by getting larger and getting stronger. But if you stop that lifting program, then you're going to lose muscle mass and you're going to lose muscle strength. And so that's involution. So that's a loss of adaptation. So this is oftentimes talked about in the concept or in the framework of Cellier and general adaptation syndrome. I won't ask you about that, but you'll see that thrown around a lot. But the idea here is that in response to a specific stimulus, the body adapts in a specific way and then is able to withstand that same stimulus being applied again. This particular graphic shows, and again, this is actually kind of a repeat of one of the graphics I showed in part 1. So there's our original level of performance here, and then we apply a training stimulus. So again, you're going to see a decline in performance, so decreased muscle strength, muscle power, for example. And then, so we're going to go route A the first time. If we just keep doing the same thing, then we sort of plateau here. But if we do, let's say another workout kind of in here, then got another training stimulus. But if we let it rest, then we're going to reach a new level of performance. So the idea on C is that we have progressively overloaded. The idea with line A is that we did just a one bout of exercise, we adapted to it, and maybe we keep doing the same thing, and so we don't make any additional adaptations. With C, we continue to progress the program in some way, increase volume, increase intensity, and so that fosters even greater adaptations, right? So if you do multiple sessions, you're going to see bigger gains in strength than you would see with just one session. And then the B line down here is we just keep grinding away without any time for rest, and so then we reach this state of overtraining, which is where you're going to be mentally fatigued, you're going to be physically fatigued, and so you just can't perform at as high of a level, and you just don't recover back to that original level. So in terms of phases of supercompensation, it's a funny term they're using here. So, you know, supercompensation means that you're able to return to a higher level than where you started. So that's the concept of supercompensation. So again, with this stuff, it's probably easiest to use strength training as an example. So the first phase, and so when we're talking about the first phase here, we're talking about this little Roman numeral 1 on this particular graphic. So in the first phase, that is in the 1 to 2 hours after the exercise. And so again, you're going to see a reduction in voluntary muscle contraction strength. There are several reasons that that probably happens. The first is reduced neural activation. Anything neural is the nervous system. So reduced neural activation. Remember that being able to increase the strength of muscle contraction, there's kind of two factors from a neuromuscular standpoint that we'll do. One is increased motor unit recruitment. So we recruit not only type 1 muscle fibers, but also type 2. And then increased rate coding, which is the alpha motor neuron depolarizes more times. So if we have a really fast depolarization rate, again, my classic example is 80 pulses or 80 depolarizations per second, and we recruit all of the motor units, all the type 1s, all the type 2s, then we're going to get a really strong, powerful muscle contraction. But if we do that repeatedly, you know, so if you did 3 sets of 10 and that weight was the most weight you could do 10 times, you're going to start to see some issues in terms of neural activation. Maybe we're just not as effective at recruiting those motor units anymore, or maybe there's a problem with the muscle cell itself. But at the very least, we are less effective at recruiting motor units after a really fatiguing activity like, you know, 3 sets of 10, 5 sets of 10, depending on your training status. Increased brain serotonin is associated with increased mental fatigue. And as I mentioned last time, you know, if you're doing 3 sets of 10, any of these types of training is uncomfortable, right? And so if you're fresh, if you're feeling good, you're willing to tolerate more discomfort. And if you're tired and not feeling very good, you're not going to tolerate as much discomfort, so you're not going to be able to express as much muscle strength. And so increased brain serotonin is associated with increased mental fatigue. Mental fatigue, again, if we're tired, we just can't endure the rigors of training. We can't produce strong muscle contractions. We're not as powerful, right? And then the other things on there, so impairments of action potential propagation due to decreased function of the SR. SR is Sarcoplasmic Reticulum. So I'm actually going to click ahead a couple slides to my little muscle cell. So hopefully this looks familiar from Structure and Function 1. So what you've got here on this graphic is this is the end of the alpha motor neuron. Remember that when the alpha motor neuron depolarizes, these little vesicles here, these little bubbles, contain a neurotransmitter called acetylcholine, which is abbreviated ACH, and you can see it right here. When acetylcholine crosses this little gap between the neuron and the muscle cell, so all this is the muscle cell, so muscle fiber, muscle cell, same thing. So when the acetylcholine crosses this little gap, it is going to bind with chemically gated channels on the muscle cell. And when it does that, those channels will open and they'll allow sodium to come rushing inside. And so then that's going to cause the inside of the muscle cell to become positive. So at rest, the inside of the muscle cell is negative. And the same thing for the neuron. At rest, the inside of the neuron is negative. So when we open those chemically gated channels, sodium comes rushing inside of the muscle cell now becomes positive. You have neighboring what are called voltage gated channels. And so as this area of the membrane becomes more positive, then the voltage gated channels open, and so then more sodium comes rushing inside. And so you get this wave of change in charge where the cell membrane goes from being negative to being positive that spreads along the length of the muscle cell. So it's going to spread this way, and it's going to spread this way. When we get over here to these things, these purple things called the T-tubules, the change in charge spreads down the T-tubules and across this blue stuff, which is called the Sarcoplasmic Reticulum. And so the Sarcoplasmic Reticulum, hopefully you'll recall that it contains calcium. And so in response to that change in charge, the Sarcoplasmic Reticulum is going to release its calcium. And so when it does, that calcium moves out of the SR, and then it moves down here amongst the contractile proteins, which are actin and myosin. And so that calcium that's been released from the sarcoplasmic reticulum is going to attach to a particular protein on actin. These little blue strings of beads, those are actin. And so calcium is going to attach to troponin, this little yellow protein here. And when calcium attaches to that, it's going to pull this other orange string, which is another protein. It's going to pull that orange string, which is called tropomyosin, off of these little binding sites, which are the little blue dots, on actin. Now when myosin, this little two-headed monster here, when myosin sees those binding sites, it's going to jump up and attach and pull actin toward the midline. And so then that's how the muscle cell shortens, is you're going to get some shortening in this sarcomere, and then some shortening in this sarcomere, and then on down, right? So as long as there is calcium being released from the sarcoplasmic reticulum, then it's going to stay bound, or it will continue to bind with troponin, which then pulls tropomyosin off of those binding sites on actin. Myosin is going to continue to attach to it, right? And so then you get continued muscle contraction. So one of the things that happens with fatigue, with repeated muscle contractions, is this process sort of goes awry. So when we want the muscle cell to relax, what we have to do is actually pump that calcium back inside of the sarcoplasmic reticulum. And so with that repeated release of calcium, pumping back in of calcium, release of calcium, pumping back in, basically we start to get kind of an equilibrium of calcium. So some of it stays out, some of it stays in, and then the sarcoplasmic reticulum is not as good at releasing the calcium. Like it does it a certain number of times, and then it just gets less good at it. And so that is important. That's an important step in muscle contraction. If the sarcoplasmic reticulum, because it's worn out, if you will, because let's stick with that. Because it's worn out, if it's less good at releasing calcium, well then the calcium can't come out and bind to troponin, and so then we can't get muscle contraction. And so you start to see with repeated muscle contractions a dysfunction of the sarcoplasmic reticulum, which then leads to a reduction in muscle contraction. If we can't release calcium, muscle cell can't contract, muscle cell can't contract, we can't produce muscular force, right? And so that's part of that whole fatigue equation post-exercise during that phase 1. So that's a long-winded explanation of this one. Increased PI. So remember that the energy molecule in a muscle cell is adenosine triphosphate, or ATP for short, and that what ends up happening is that we break that molecule. So we use an enzyme called ATPase, and we split the last phosphate off of ATP, and so then we're left with adenosine diphosphate, or ADP, and then a free phosphate, and so that's indicated by a little PI. And so that presence of increased free phosphate may reduce the calcium sensitivity inside of the myofibril. So that has some interaction with the sarcoplasmic reticulum, and so it again is going to play a role in hampering that release of calcium from the sarcoplasmic reticulum. And then we're going to start an inflammatory process. So inflammation is going to cause swelling, going to cause pain. Typically you don't see pain within the first couple hours after exercise, but that swelling of the muscle cell may, in the short term, play a role in reducing our ability to voluntarily contract the muscle. We're also going to see a reduction in substrates. And what are substrates? So a substrate is a fuel, right? So we have different things that we can use to remake ATP, because again, ATP is the energy molecule. And we've got macronutrients that we can break down to remake ATP. So we got carbohydrates, fat, and protein. And so all of those, carbohydrates, fat, and protein, as well as ATP and phosphocreatine, would be substrates. They'd be energy sources. They'd be molecules whose bonds we can break to help us remake ATP. And so with a fatiguing exercise bout, you're going to get a reduction in glycogen. So remember that glycogen is sugar stored inside of the muscle cell. And so if we've reduced our sugar content inside of the muscle, we just have less fuel to remake ATP. And if we can't remake ATP as efficiently, well, then we can't drive muscle contraction as effectively. So that reduction in glycogen is going to result in a reduction in muscle strength, and in particular, a reduction in muscle endurance. And then the last thing on there, the PCR, stands for phosphocreatine. You'll also see it as big C, little r, big P, so creatine phosphate, either way. But I always write it PCR, or it'll always be in these slides as PCR. So phosphocreatine is the most rapid rebuilder, if you will, of ATP. So phosphocreatine can give up its little phosphate, its P there. It'll give that up, and then that will push adenosine diphosphate and a free phosphate back together, and now we've remade an ATP molecule. But creatine now has to go find a phosphate, so we have to rebuild the creatine. So if we reduce fuel sources inside the muscle cell, so if the muscle cell's fuel tank is empty or low, it just can't contract as strongly, or it can't contract as long. So all of that stuff happens in response to that initial training session, and all of that stuff results in a reduction in muscle strength, muscle power, and muscle endurance. During phase 2, so going back to that graphic, so here's phase 2. So now we're in the compensation phase, so we're rebuilding is kind of a way of thinking about this. So that's the next 24 to 48 hours. And this depends upon the intensity of the exercise, the training status of the individual. There's a number of factors that play into this, but generally speaking, it's somewhere in the neighborhood of 24 to 48 hours. And so what we're going to do then in those 24 to 48 hours is we're going to resynthesize or remake ATP. So after repeated muscle contractions, you're going to have a lot more free phosphates in the muscle cell and a lot of ADP, adenosine diphosphates, floating around the muscle cell. We got to rebuild those if we want the muscle cell to recover and to be able to contract just as strongly. So that's part of the equation. So we got to rebuild the ATPs. We also have to rephosphorylate or reattach the phosphates back to creatine to remake creatine phosphate or phosphocreatine. Again, either way. So we're going to rebuild all those. And so that process happens aerobically, and we'll talk about that more in a minute. But that's going to be, you're going to use the aerobic system to reset the anaerobic system is kind of a way of thinking about it. MVC stands for maximum voluntary contraction. Maximum voluntary contraction is how hard you're willing to contract the muscle. And so that's partially restored. You'll see some restoration of muscle strength, not back to your original levels, but better than you were a couple hours after exercise. We're going to resynthesize glycogen, so we are going to rebuild our glycogen stores. So you're probably at least familiar with this concept in terms of, after exercise, oftentimes people will drink maybe chocolate milk, or they'll drink some kind of a recovery drink, right? So you may drink some combination of protein and glucose. And so what you're doing, even if you don't think about it in this fashion, but what you're doing is you're trying to supply sugar to allow your body to move that sugar, that glucose, inside of the muscle cells to resynthesize, to remake your intramuscular stores of carbohydrates, aka your glycogen stores. So we're going to resynthesize or rebuild your glycogen stores within 20 to 24 hours. And another factor here is it depends upon what you eat. So if you don't, consume carbohydrates, well then you're not going to resynthesize your glycogen nearly as readily as you would if you consume carbohydrates after exercise. We're also going to see excess post-exercise oxygen consumption, abbreviated Epoc, and this is one of those sort of complicated topics that we're going to talk about today. And then you're going to see increased protein synthesis. So protein synthesis is making new proteins. And so when we're making new proteins, depending upon the type of training, maybe we're making more actins and more myosins, which are the contractile proteins. So we're especially in response to strength training, we're going to eventually make the muscle cell larger. And we make the muscle cell larger by adding additional actins and myosins, the contractile proteins. And the more contractile proteins you have, the stronger muscle contraction you're going to get eventually. So that's part of an increase in protein synthesis. That increases 50% in the first 4 hours after exercise and increases 100% in the 24 hours after exercise. So you double your protein synthesis rate in the 24 hours after exercise. So not only are we rebuilding the contractile proteins, but if you did high volume strength training, if you did endurance exercise, you have to make more mitochondria, right? So there's a lot of phospholipids in mitochondria, but there are some proteins involved in that, and so increased protein synthesis is related to building more mitochondria, and we'll talk about why that's important. Rebuilding proteins that we need inside of the membrane or in the membrane of the muscle cell, right? So protein synthesis means that you're building proteins to try to repair the muscle or to try to, in the case of adding more actin and myosin, to improve the capacity of the muscle. All right, now, so let's talk Epoc. So we should probably just go through the term first so it's not quite as intimidating. So excess, too much of, or in this case just more than, post-exercise, after exercise, oxygen consumption, how often you're breathing, it's related to that, right? So all this really means excess post-exercise oxygen consumption is that after an exercise bout, you are consuming more oxygen, you're breathing more times per minute than you were before the exercise. And so what that indicates is that you are recovering. You are trying to rebuild proteins, you are trying to reset those ion gradients. So your charged particles are your ions, so things like calcium, potassium, sodium, etc. We're going to have to pump those back to where they belong. So we'll have to pump calcium back inside of the sarcoplasmic reticulum. We'll have to pump sodium back outside of the muscle cell, pump it back outside of the neurons, pump potassium back in. And so all of that takes energy. And so we're going to use oxygen to make ATP molecules, or to remake ATP molecules more accurately, in order to do all of those things. So it means that our metabolism is working at a higher rate after the exercise than it was before the exercise. So that's what Epoc is as a concept. So I'm going to show you a couple graphics related to Epoc. And there's a term on here which is oxygen deficit. So you see O2 deficit, that's oxygen deficit. And so what you've got here is that somebody starts exercising, and this is pretty light exercise, as you can see up top, light aerobic exercise. This is actually from a different textbook. This is from an exercise physiology textbook I've used in the past. So if you were to try to find it in the periodization book, it's not in there. But at any rate, so somebody engages in light aerobic exercise. So let's say they just walk, right? So at a fairly leisurely pace. So they had been sitting, they had been sedentary, and they get up and they start walking. Well, in walking, you're going to have higher energy demands than you had when you were sitting. And so we have to supply more oxygen to remake more ATP because the energy demands are higher. And so what's going to happen then is that when we transition from sitting to walking, well, now we have to direct more blood flow to the working muscles, right? And so we have to, for example, at rest, in your calf musculature, only 1 out of every 30 or 40 blood vessels is open. We don't have to have blood flow going to all of the muscle cells all the time. But when we start walking, we're going to use those calf muscles. They're going to be contracting, and so they're going to need oxygen to make ATP. So we're going to have to open up blood vessels to the calf musculature. We'll have to open up blood vessels to the quads, to the glutes, to the hamstrings, etc. And then as a consequence, we're also going to close off blood vessels in the abdomen, so to the kidneys, to the liver, intestines, etc. And so all of that takes a little bit of time, right? So directing blood flow away from the abdomen and toward the working musculature takes a little bit of time. And not only do we have to direct blood flow to the working muscle cells by opening up blood vessels, but we also have to increase our heart rate and increase how hard the heart is contracting. So that is increasing blood pressure to get more blood flow to those working muscles. And so there's a lag time. It takes a little while for all those things to happen, right? But those working calf muscles, in that example, need energy. They need ATP right now. So they don't have the oxygen to meet the energy demands, but they're they have to use ATP right now to fuel muscle contraction. And so what can they do before they receive enough oxygen that still allows them to contract? Well, they can use anaerobic sources. And so they can use the ATP phosphocreatine system, which I'll talk about more here in a second. And they can use the glycolytic system. So they can break down phosphocreatines to remake ATP in one step that doesn't require any oxygen. And they can break down glycogen. They can break down sugar inside of the muscle cell. 10 steps, but none of them requires oxygen either. And so what they do then is they can use that anaerobic system kind of like a credit card. So they can or generate energy before they get the oxygen to actually produce the energy. They're spending before they get the paycheck, if you will, the paycheck being the oxygen in this example. And so what we're doing there then is we're running up a deficit. And so this green area here is that oxygen deficit. So if they had been able to supply oxygen to the working muscles right from the start of the exercise, we needed oxygen at this rate, but it took us a little while to open those blood vessels and to increase our heart rate, right? And so this area then here, all this green, is anaerobic energy. So we have borrowed against our credit card. We have broken down phosphocreatine and we've broken down glycogen anaerobically to supply that energy. And then what we're going to do is eventually we're going to be able to supply enough energy aerobically. And so now our aerobic energy demands or energy supply meets the demands. Right, so we're not doing aerobic energy production anymore. We're not doing anaerobic energy production anymore. Everything's aerobic. And then after exercise, so after we stop and we sit down, then our heart rate will stay elevated very briefly after the exercise, and our breathing rate will stay elevated a little bit after the exercise. And so that is our excess post-exercise oxygen consumption. So that's this yellow area here. And so what we're doing then is when we're resting, we are remaking all of these phosphocreatines and getting rid of the lactate that we generated when we were working anaerobically at the start of the exercise. So had to use some energy anaerobically over here, and then we replace it over here. So we rebuild those things. So that is your excess post-exercise oxygen consumption. So that's part of the equation. And so in light exercise, that's basically all you're doing. So if you walk, across campus or something, the excess post-exercise oxygen consumption, you're not going to be breathing heavy, probably. When if it's just a leisurely walk across campus, when you get to your next class, you're not going to be breathing heavy when you sit down. You may have elevated breathing for a second or two, but it's not like heavy breathing, right? And so you're able to remake those phosphocreatines, remake the ATPs, and then get rid of any lactate that you produced aerobically at that time during that recovery. So as I was saying that, it was occurring to me that I should probably explain what's happening with lactate. So when you break down glucose, break down sugar anaerobically inside of a muscle cell, what ends up happening is that the byproduct of doing it not in the presence of oxygen is lactate or lactic acid. And so what we're doing on that green side then, or under the green curve, if you will, is producing lactate and running through our phosphocreatine and ATP stores. So that's light exercise. What happens if we, let's say, do repeated sprints? Well, we can actually work above our aerobic capacity. So there's a term on here that you've probably heard. So aerobic capacity and VO2 max, those are the same things, right? So sometimes you'll see one, sometimes you'll see the other, they're the same thing. And what they deal with, broadly speaking, is our ability to supply and use oxygen. So from a supply side, we have to be able to, you don't see any adaptations in the lungs to endurance training. So the lungs don't change, but the heart changes with training. So we have to be able to pump the blood, right? So that's the job of the heart. The blood has to be able to hold enough oxygen. So one of the adaptations we get with endurance training is a little bit higher blood volume. So you have more plasma initially, and then after about a month of training, you have a few more red blood cells. So we got to be able to transport oxygen, which is the job of the heart and the blood. And then at the level of the working muscle, we have to be able to use that oxygen. So we have to be able to get it inside of the muscle cell, which requires capillaries. And not only do we have to get that oxygen inside of the muscle cell, we have to get it to where we need it, which is actually inside of the mitochondria. And so for most people, their VO2 max is somewhere in the neighborhood of the fastest pace you could run a mile. That's about as fast as you can supply energy aerobically. Although if you're an elite miler, you're obviously working over your VO2 max. But just to give you kind of a ballpark of what your VO2 max is. So the VO2 max then again deals with your ability to supply from the cardiovascular system standpoint and use from the muscular system standpoint oxygen. So you can work at a higher level than we can supply and use oxygen. So if you're sprinting, there's no way your heart could ever supply enough oxygen or your muscles could ever use enough oxygen to keep up with those energy demands. The energy demands of sprinting are so high that we have to use anaerobic sources. Because on the anaerobic side, so we'll talk about this here in a second, but on the anaerobic side, the anaerobic sources are ATP, phosphocreatine, and then the glycolytic system. So breaking down glucose, glycogen, not in the presence of oxygen. And so those things like the ATP phosphocreatine system has 1 step to remake an ATP. The glycolytic system has 10 steps to remake an ATP. And then on the aerobic side, there are somewhere in the neighborhood of 20 or more steps, right? And so the more steps you add, the more time it takes. And so the energy transfer on the aerobic side, so with oxygen, is just much slower than it is on the anaerobic side. So when we're doing things like sprinting, lifting heavy weights, those are kind of the big ones, jumping. When we're doing those things, the muscles are running through ATP faster than we could ever supply or use oxygen inside of those muscles. So we're operating then above our aerobic capacity, above our VO2 max. And so that's what's depicted here. So this is, let's say, somebody who is running, I don't know, we'll call it a 400 meter dash or an 800 meter, something like that. So they're going to take off running. And so again, we have to open up blood vessels in the calves, in the quads, hamstrings, etc. We have to increase heart rate. We have to increase blood pressure. And so all the while, those muscle cells are still contracting. And so they're still burning through ATP, but they're burning through it, or they are rebuilding their ATPs anaerobically. So they're breaking down glycogen, they're making lactate, they're breaking down phosphocreatine. And so even when they reach steady state, they don't ever reach steady state here, sorry, not to throw you off. Even when they plateau in their speed, so this is as fast as they're going to run this race, they're still operating above their VO2 max. So they are generating lots of lactic acid, right? So if you ever run a 400 or an 800, stick with an 800, you know that it burns lots and lots. And so that burning sensation is that accumulation of lactate is kind of the most straightforward way to think about it. So generated lots of lactate, run through lots of phosphocreatine. And then we cross the finish line, and the athlete is going to be just huffing and puffing, right? Just breathing all kinds of heavy. And what you're going to see is because they worked so high above their VO2 max, it takes them much longer to recover. So that recovery rate, that excess post-exercise oxygen consumption, which is the yellow stuff, that may last 24, 48 hours. In some cases, 72 hours, right? So the more intense the exercise, the longer the excess post-exercise oxygen consumption. So they're not fully recovered until that epoch goes away, until their oxygen consumption returns to normal. Why does that matter or why would you be interested in that? During that phase of epoch, during that recovery, you're burning a lot of extra calories relative to what you did before the exercise, right? So if you lift weights, you don't burn many calories actually lifting the weights. You burn most of your calories in the 72-ish hours after the exercise when you are rebuilding the muscles, when you are pumping calcium back into the sarcoplasmic reticulum, when you are rebuilding your ATPs aerobically. So in that recovery then, you are using a lot more calories, right? And so from a physique standpoint, that's why you might be interested in that. But from an athletic recovery standpoint, when they're here in this epoch, they are not quite recovered yet. So epoch then, excess post-exercise oxygen consumption, it is trying to recover from using the anaerobic energy system. And then, because there's a couple components to this, and I don't want to get into that much detail, but not only are you rebuilding your ATPs and phosphocreatines aerobically, but the other thing that you're doing is obviously during exercise you have so hormones associated with that. So you have epinephrine, norepinephrine. Those are going to raise your metabolic rate for a little bit after the exercise. Your core temperature is probably higher, so it takes a little while for that to come back down. Like I mentioned, you have to pump your ions back to where they belong. That takes energy. Some of your other hormones, like growth hormone, are going to affect your rate of fat breakdown, which is called lipolysis, in the hours after exercise. So with more intense exercise, then you get a bigger change in that hormonal milieu, that mix of hormones, and so then that lengthens this recovery phase. So that's Epoc. So as it pertains to what we're talking about, Epoc is going to last, depending on the intensity of the exercise, 24 to 48 hours, potentially as long as like 72. So as far as training theory goes, if you're familiar with the idea of high intensity interval training or HIIT training, there's a bunch of different ways that you can do it, but like one example of doing high intensity interval training would be to do like Tabata protocols, where you're doing like 20-second sprint, 10-second rest, 20-second sprint, 10-second rest for, let's say, 8 repetitions, or a set of 8. That type of training increases your metabolic rate during the recovery period much more than if you had done the same volume of work at a much lower intensity. So if you'd done the same, the Tabata thing takes, give or take, like 4 minutes. So if you had just done 4 minutes of moderate intensity exercise, you're going to recover from the moderate intensity exercise pretty quick, because from this standpoint, you didn't get too far above your VO2 max. Maybe, actually in that case, you wouldn't get to your VO2 max. So you never got there, so you just have this initial oxygen deficit. But if you're doing those repeated sprints, well, then you are working above your VO2 max, and so you end up with that elevated metabolism. So that's one of the things that you see a lot around, or in the exercise literature now, is talking about the utility of short, really high intensity sprint type things from a calorie burning standpoint. And that's why, is because of the Epoc, because it takes so long for you to return back to your resting state, again, maybe as long as 72 hours after the initial bout of exercise. All right, and then phase 3. So now we're into supercompensation. So, 2-3 days after the exercise. So, and again, this varies depending upon the intensity of the exercise and the volume. So one of the terms you're going to see a lot coming up is this concept of load. And so load is the combination of intensity, volume, and frequency, right? And so depending on the load of the exercise, that combination, particularly of volume and intensity, you may not reach supercompensation for as long as a week. But in general, it takes about 3 days. So I say that, for example, if you've ever done, maybe you got fairly deconditioned and you're foolish like I am and you haven't lifted weights in a while and you're like, I'm going to do like I used to do and do like 5 sets of 10, 12, whatever, really high volume stuff. And then you're sore for 7 days after, right? So in that example, because I tend to do that all the time, been like, I haven't lifted in a while, I'm going to go back and lift, but I'm going to do like I always do. I'm going to make the most of my time and do 5 sets. And then I walk funny for a week. I wouldn't be in supercompensation on day 3. I'm still pretty sore at day 3, so I'm not stronger on day 3 than I was on day 1. So the supercompensation in that case takes a little bit longer. It's, like I said, it's more like a week. But typical workouts, somewhere between 3-4 days, and then you're in that supercompensation phase. But as you can see, if we don't apply another exercise stimulus when you're in that supercompensation phase, so you made some adaptation, you're then going to lose those adaptations. So that's, again, involution is the term for that. So what we want to do then is train frequently enough that we catch the athlete when they've recovered, we apply another exercise stimulus so that they don't lose what they've gained, and then we build on that new base that they've established. So we build on that supercompensation phase. And then eventually that becomes a noticeable increase in strength, a noticeable increase in power, right? So they can add 10 pounds to their bench press. They can jump a 1/4 inch higher on their vertical jump, those kinds of things. So then it becomes noticeable and measurable. So that's the relevance here in phase 3. Other things, psychological supercompensation. What they're getting at there in the textbook, I think, is the idea that, if you've done, let's say, a really hard workout, well, now you know you can do that one. You got through it and you adapted. And so you can do a little bit more because you know you got through it last time. And so I think that's what they're getting at with psychological supercompensation. And then the last thing on there, you can see the glycogen stores are fully replenished. So the muscles are holding as much glycogen as they can. Actually, in terms of supercompensation, so, supercompensation is you're at a higher level than you were before you started the exercise. One of the things that happens with both sprint training and endurance training in the trained muscle cells is that they store more glycogen, because we use glucose to fuel endurance exercise, but we also use glucose to fuel sprint type exercises. And so for both, you're going to see increased glycogen content in the trained musculature. So if you're familiar with, at least in my experience, being around like really recreational level runners, oftentimes they're like, oh, we got a race on Saturday, we got to go carb load on Friday. And so basically what they mean is they're going to go to the Olive Garden or something and just like load up on pasta. And so that's not really how carb loading works. There is actually this, truly how carb loading works is there's a depletion phase. It takes about a week. You have to like go low carb for several days, train at a really high intensity, and then you taper the intensity and the volume of the training, and then increase your carbohydrate intake. Recreational athletes don't do that, but they understand, or they've heard that carbohydrates fuel aerobic exercise or endurance exercise, and so they're like, well, we're going to go carb load, we're going to go consume a lot more carbs. And they will store a little bit extra, but once their carbohydrate stores are full, they're full. But again, after training, after training for 4, 8, 12 weeks, you're going to see in the trained muscle cell, it can store more glucose, more glycogen than it could before the training. So you're going to see a supercompensation in terms of glycogen stores. And so in terms of phase 4, so again, if we don't apply another stimulus here, they're going to lose the adaptations that they made. So one of the things that you can see from this graphic, which this is kind of a weird graphic, but the gist of it is, so we have fatigued ourselves or fatigued our athlete, they've improved, they're starting to come down or get to involution, so then we train them again, supercompensation a little bit, they're starting to come back down, train them again. And so the idea here is that if we can squeeze together more frequent training sessions, so rather than like every 3rd day, if we can get them to do every 2nd day or something like that, again, depending upon the intensity and the volume of the exercise, the more training, the better within limits, right? So we want to avoid overtraining, which is where they just, again, they're mentally and physically fatigued. So we have to allow, it's this interesting balance of allowing recovery, but not letting them rest so long they start to lose some of their gains. So again, typically you want, at the very least to be training, give or take, every 3 to 4 days, which is pretty spread out. Most of our athletes at the college level, for example, or even at the high school level, work out 4 days a week, right? So they may work out Friday, they're off Saturday, Sunday, they work out again on Monday. So there's only, 2 days there. So they don't have time to really lose anything. You see athletes, especially in endurance sports, who will do, multiple sessions in a day. So you'll see they'll do, cross country, sometimes they'll do morning runs and afternoon runs. But the key there is that they change the volume and the intensity of the workouts. So maybe they have their hard workout in the morning and then they're just doing miles in the afternoon. So one of the things about endurance athletes is typically, at least as far as runners go, they usually only have like 2 to 3 like hard or what they sometimes refer to as quality workouts per week. So there's a lot of miles oftentimes, but there's not necessarily a lot of like really, really difficult miles. That's only 2-3 days a week for many of them. Because if you do more than that, well, then that's overtraining and then they become stale and all of that, more likely to get injured. All right, so a lot of the stuff we've already covered, so that works out really well. But So in terms of sources of energy, remember that adenosine triphosphate, or ATP, directly fuels muscle contraction. In terms of what's going on inside of the muscle cell, so we went through all the calcium release, actin, myosin, etc. So remember that when the binding sites on actin are exposed, myosin is going to jump up and attach and pull it to the midline. To reset myosin, we have to break down an ATP. And the energy that's given off when we break down ATP allows us to pull the myosin head. So here's myosin pulled toward the midline. If we break down an ATP now, that'll allow us to detach myosin, so the actual order here. We attach an ATP to this myosin, so then it detaches from actin. Then we split that ATP. So you have the enzyme on the heads of the myosins called myosin ATPase. So we split that ATP. The energy given off from splitting that ATP returns the myosin back to its original position, which is up here. So it's called cocking the myosin head. So we use ATP to reset the myosin head. So one of the ways I always describe myosin at rest is it's like a coiled spring or a cocked spring, right? So it's ready to go, and as soon as we release it, it jumps up and attaches to the actin. And then we use energy, ATP, to reset it. So that's how ATP drives muscle contraction, is it resets those myosin heads so that they can uncoil like a spring. So that's our first energy source. Then, so what we have to do, if we want the muscle to keep contracting, is we've got to make more ATP or remake more ATP. So I keep saying remake because we basically push the ADP, the diphosphate, and then that free phosphate back together. So we remake ATP, we break it down. We remake it, we break it down. There are, when you get to XPhys, you'll talk about like AMP and some other things, but just the easiest way to conceptualize it is break it into ADP and free phosphate, put them back together. Break them down, put them back together, and just do that over and over and over again. So we can remake ATP anaerobically, so not in the presence of oxygen. We can use phosphocreatine or the glycolytic system to do that, or we can remake ATP aerobically, so in the presence of oxygen. All right, so as far as the anaerobic sources go, so again, I've kind of already covered this, but this is the ATP phosphocreatine system. And so you've got your little creatine phosphate here, your ADP. So we got to rebuild this. We got to transform this adenosine diphosphate into an ATP. And so what we'll do is this creatine phosphate, ADP will come together. The enzyme is called creatine kinase. And then what we're left with, so basically we'll migrate this phosphate from here to here. And then what we're left with is an adenosine triphosphate. You can see its energy as symbolized by its little sun-looking thing there. And then a free creatine. So in one step, we have a recreated ATP. Now you might think to yourself, like, why don't we just store a boatload of phosphocreatine? And the answer is because creatine phosphate or phosphocreatine is a really heavy molecule, and it tends to pull a lot of water with it inside of the cell. So for example, when people supplement with creatine monohydrate, so you add a bunch of creatines to the muscle cell, we phosphorylate them, and then what you'll see is that typically if they load it, so if they take 20 grams a day for a week, so you take 4 or 5 gram servings over the course of every day for a week, at the end of that week they've usually gained about 5 pounds. Depends on their initial starting weight, but typically they've gained about 5 pounds. And the reason they've gained 5 pounds is because since they've stored or they've saturated the muscle cells with all that creatine phosphate, it pulls water with it. And so they've gained 5 pounds of water weight. So we can't store too much ATP or too much phosphocreatine because both of them are really big, heavy molecules, and then they tend to pull water. And so if we stored lots and lots more ATP, we'd all be like big water balloons. So we have to have a much more efficient way to remake ATP rather than just storing a bunch of ATP. So the ATP phosphocreatine system then, one-step reaction, one enzyme, creatine kinase, and we got a new ATP. But it is very limited by our ability to store phosphocreatine in the muscles. So the ATP phosphocreatine system supplies all-out energy for about 10 to 15 seconds. So all-out sprint, 10 to 15 seconds, so give or take a 100-meter dash and less, that's pure ATP phosphocreatine. Then if we go a little bit longer, so let's say we go from 100-meter dash to 200 meters, 400 meters, right? So we're going half a lap around the track instead of a straightaway, or a full lap around the track as fast as you can. Well, on that initial part of the sprint, you've worn out your ATP phosphocreatine system. You've already broken down all the phosphocreatines. And so now we got to shift gears. And the gear that we shift to is the glycolytic system. And so what we're going to do again is to break down glucose, glycogen, inside of the muscle cell. And we're going to do that in 10 steps. And at the end of those 10 steps, what we're left with are 2 ATPs. So that's a lot of steps for 2 ATPs. And pyruvic acid, or pyruvate is another way you'll see that. So if we're exercising at a really high intensity, so again, if you're running a 400-meter dash as fast as you can, that is, you're running above your aerobic capacity. You're working at an intensity that is so high that you can't possibly supply and use oxygen at that rate. What we can do is to, or what we're going to do is to generate lactic acid. So pyruvate gets turned into lactic acid as a temporary storage site. So it's not depicted in this diagram. See if I have it in these other ones. I don't. So what ends up happening during all of these 10 steps of glycolysis is that we pull hydrogens off of these intermediaries of glucose. And those hydrogens we have to get rid of, because otherwise the inside of the muscle cell becomes too acidic too fast. Now obviously it's still going to be acidic, but it's more of a problem if you just release a bunch of free hydrogens. So what we do then is we take those hydrogens and we actually attach them to pyruvate, and then pyruvate becomes lactic acid. So lactic acid is pyruvate with some added hydrogen. But lactic acid, still an acid, so we got to get rid of it. And so what we'll do, you can see here, you can release it in the blood, and the heart can use it, or the heart's really good at breaking down lactic acid, or when the intensity of the exercise slows down, so if you sprint and then do slow jog for a little bit, now the aerobic system gets to catch up, and so we can supply more oxygen to the muscle, and so we can pull those hydrogens off of lactic acid and take them to the mitochondria. So that's what happens there. But in the anaerobic system, key takeaways here, or in the glycolytic system, sorry, key takeaways, it is anaerobic, so we don't need oxygen. In 10 steps, we can generate 2 ATPs. We also generate 2 pyruvates, and then 2 NADHs, so those are the 2 hydrogens I was talking about. They're attached to NAD molecules. And so the glycolytic system is limited by the pH of the muscle cell. So if you generate lots and lots of lactic acid, the pH of the cell drops a bunch, it becomes acidic on the inside of the cell. That interferes with the contractile machinery of actin and myosin. And so then because of that burning, because of that acidity inside the cell, eventually you're not going to be able to contract it anymore. So the glycolytic system is limited by its byproducts, primarily. It's also limited by stores. You can store about 2,000 calories in the glycolytic system. So that's enough energy to run approximately 20 miles. So if you run a marathon, you're going to run through all your glycogen. And so you either have to, you have to kind of do a combination of you run a little bit slower so you can supply energy aerobically, but also you add supplemental glucose. So you eat, jelly beans and gummies and Gatorade and all that stuff during the course of the race. So those are the fastest 2 ways to remake ATP. 1 step phosphocreatine, 10 steps glycolytic. Phosphocreatine can't store much. Glycolytic system generates lactic acid, so it's limited by the acidity. The reason my little muscle cells on here, so all of the anaerobic stuff happens out here in the cytosol, which in a muscle cell is called the sarcoplasm. So everything anaerobic happens out here. Everything aerobic happens inside of the mitochondria, which hopefully you'll remember from your old biology classes as the powerhouse of the cell. The reason the mitochondria is the powerhouse of the cell is that's where we make the vast majority of our ATP, and we do it aerobically. So that's where the oxygen has to go that we've delivered to the muscle cell. We have to shuttle it from, this one doesn't have capillaries, but we have to shuttle it from the capillary to the mitochondria. We'll come back to lactate threshold. All right, the aerobic system. So the aerobic system, again, happens inside of the mitochondria. And so what we'll do from the standpoint of glycolysis, so that pyruvate, we'll actually move that pyruvate, which you can see on this graphic. We'll move that pyruvate inside of the mitochondria. Once it's inside of the mitochondria, it gets converted to acetyl-CoA and then runs through these 11 additional enzyme steps. And so as it runs through there, we generate more hydrogens. So here's one, here's one, here's another one, here's another one. So we generate more hydrogens, and then there's one up top too. We generate more hydrogens as part of that process. And so again, what we have to do is to get rid of those hydrogens. And so what we'll do, so all of this stuff, this is the citric acid cycle, also known as the Krebs cycle, that happens inside of the inner mitochondrial membrane. So as I'm saying that, I'm going to back up a step. So your little mitochondria here, you got two membranes. There's an outer membrane here, inner membrane here. So all of the stuff I just showed you, the citric acid cycle, or the Krebs cycle, where we convert pyruvate to essentially CO2 and a bunch of NADHs, so, and FADH too. So all that stuff, the citric acid cycle or the Krebs cycle, happens inside this inner membrane here. And then what we're going to do is we're going to take all those hydrogens, and now we got to get rid of them. But when we get rid of them, they're going to go through what's called the electron transport chain. What we're going to do is the NADH that was made over here by stripping a hydrogen, in this case off of isocitrate, that NADH is going to drop off its hydrogen. We're actually going to pull electrons off of it, and we're going to pump that hydrogen out here into this intermembrane space between the outer membrane and the inner membrane. And then those electrons are going to shuttle between these cytochromes, and they lose energy as they do that. And so as they lose energy, that's what allows us to pump these hydrogens out here. And so you get in that intermembrane space, you get this area with lots and lots of hydrogen. And so what we'll do then is hydrogen wants to move back with its concentration gradient. It wants to go from an area of high concentration, so in the intermembrane space, to an area of low concentration, which is here inside this inner membrane. And so what we'll do is we'll actually let hydrogen come back inside of that inner membrane, but when it does, when it moves with its concentration gradient, that movement of hydrogens actually spins this protein called ATP synthase. These spin pretty fast, as many as 50 times a second, so it's a really fast moving protein, but when these spin, they rephosphorylate, so add a phosphate back to ADP. So we got an ADP, free phosphate. As hydrogen comes through, it spins ATP synthase, which puts those two back together, and we have remade our ATP. But now that hydrogen's back inside, we got to get rid of it. And so what we'll do is we attach it to, actually two of them, we'll attach to oxygen, and we get H2O. And so the way that we get rid of those hydrogens using oxygen is to make what's called metabolic water. So we use oxygen to bond it with two hydrogens to make H2O, and then we get rid of those hydrogens that way so that the inside of the cell doesn't become too acidic. So that's the aerobic side. So that's where oxygen comes into this process, is oxygen helps us to get rid of the hydrogens. But as you can see, so if we stick with the carbohydrate example, breaking down carbohydrates aerobically takes a lot of steps, right? So there were 10 steps involved in breaking down glucose or glycogen as far as going through glycolysis. Then we end up with pyruvate. Then we got to transport pyruvate inside of the inner membrane of the mitochondria. It goes through 11 more steps. Then we take the hydrogens and we run them through the electron transport chain, which is at least 5 more steps. So those steps add up in terms of time. And so the aerobic system then can produce a lot of energy, but it takes it a long time. So when we break down a glucose molecule anaerobically, so we're just going through glycolysis, we get back 2 ATPs from it. When we break it down aerobically, so when it runs through the citric acid cycle on the electron transport chain, now we get back 32, right? So we produce way more energy using the aerobic system, but it just takes a long time to do it. So aerobically, we just can't work at as high of an intensity. And again, the aerobic side is also limited by our ability to deliver oxygen to the tissues and to use it at the tissues. So we actually have to get it from the bloodstream into the mitochondria, and that takes time too. So in terms of, so the way I've described this, and kind of the easiest way to conceptualize it at the outset is like you're shifting gears, right? So if you are sprinting, you're ATP phosphocreatine. If you are like a middle distance, now you are ATP phosphocreatine, glycolytic. And then if you're going more than 90 seconds, maybe 2 minutes, now you're aerobic, right? And so I've kind of described it as shifting gears from ATP phosphocreatine glycolytic to aerobic. But the reality is you're trying to use all the systems at once, but again, it just takes a little while for the aerobic system to get ramped up. So in terms of the overlap of the two, if we're doing 10-second sprint, so 90% of the energy, 98%, close to, comes from anaerobic sources, so ATP phosphocreatine system. 30-second sprint, so now you're running something like a 200-ish. Not if you're good, but if you're slower, you're running something like a 30-second 200-ish. And so now we're at like 70% so one of the things I should call your attention to here in looking at this graphic, so there's actually two things on this graphic. Where it says aerobic in blue, that should be anaerobic. I just now noticed that, sorry. But it's not my fault. Textbook company screwed this one up. So this blue is anaerobic, even though it says aerobic on the key. The gray is aerobic, even though it says anaerobic on the key. So in our 100-yard dash or 100-meter dash, 90% of the energy comes anaerobic. Disregard the key. The blue is anaerobic. If we're running our 200, 70% is anaerobic. Where it says 60 minutes should be 60 seconds, because again, they weren't real careful making this graphic. So if you're running, give or take a 400-meter dash, now you're in about 50-50. And then going all out for 2 minutes. So now maybe you're at an 800 if you're a really good runner. So now you're at 40%, right? And so the longer we go, so now the aerobic system is starting to catch up, and our intensity is lower, right? So we're running slower. Certainly you run an 800 meter a lot slower than you run a 100 meter, right? And so as the intensity drops and we add time, the aerobic system is able to catch up. And so it contributes a larger and larger component the longer we go and the lower the intensity drops. If you're going for 2 hours, that's almost entirely aerobic. So that's what you got pictured there. So key points, and then we're done. So for most cellular processes, including muscle contraction, the energy is supplied by breakdown of ATP. In order to continue activity like muscle contraction, we have to reproduce or rephosphorylate ATP from adenosine diphosphate and free phosphate, so ADP and PI, and we can do that with or without oxygen. So our anaerobic sources then of ATP rephosphorylation, which again just means to rebuild ATP, include the phosphocreatine and the glycolytic system. So the phosphocreatine system, one-step reaction, but we're limited by our supplies, but it supplies all-out energy for 10 seconds. Glycolytic system, 10 steps. Lactic acid or lactate is going to limit use, but it's going to supply all-out energy from somewhere between 90 seconds and 2 minutes. Anything longer than 2 minutes, now we have to remake ATP aerobically. And so what we do then is we break down carbohydrates, we break down fat, and we break down protein, primarily carbohydrates and fat, to remake ATP using oxygen. And so the key takeaway there, again, takes a long time and there's a lot of steps. So the energy transfer rate just isn't as high on the aerobic side. From a training standpoint, all of those energy systems improve with training, right? So anaerobic training, so sprint type training, lifting weights would also be anaerobic training, increases the intramuscular stores of ATP, phosphocreatine, and glycogen, and the enzymes to break them down. So it increases the number of creatine kinase enzymes, and then it increases those 10 enzymes that are involved in glycolysis. Aerobic training goes the other way to some extent. So aerobic training, we're going to add mitochondria, because that's where we use the oxygen to the muscle cell. So we get more mitochondria, we get larger mitochondria. We also add capillaries to the trained muscle cell, or the muscle fiber. So after or before training, an individual muscle cell may have 3 or 4 capillaries. After training, it may have 7 or 8. So you can double the number of capillaries after training. Not after one session, right? So over months and years, you can double the number of capillaries. But what that means then is with more capillaries, you got more blood flowing past the muscle cell, which means you can supply more oxygen to the muscle cell, which then you can use inside all those mitochondria you built, right? And so in addition to having more and larger mitochondria, we also have more aerobic enzymes, also known as citric acid cycle enzymes. So the enzymes involved in that 11 steps where we break down pyruvate into acetyl-CoA and ultimately into things like NADH, FADH2, and carbon dioxide. Those are the enzymes involved in that. So why do you care? Because of specificity of training. You can't maximize all the energy systems. You can't maximally improve your aerobic capacity and also maximize your anaerobic energy transfer. So that's why we care. So especially if you've got, like I said, a sprint athlete, if we do a bunch of aerobic training with them, we're going to make a bunch of adaptations that are good for aerobic training, but actually bad for anaerobic performance, right? So that's where some of that problem comes in. Same kind of thing, but to a lesser extent. If we do a bunch of sprint type training with an endurance athlete, we're going to make adaptations that are not as useful for our endurance type athlete. So you can't maximize all the systems at one time. There was one thing, now that I'm thinking about it, that I need to come back to. This concept of lactate threshold. And so lactate threshold is where you start to accumulate lactate. And so lactate threshold, the easiest way to think about it is it's the changeover from aerobic energy production to anaerobic energy production. So if we go out and you, let's say, jog a mile, and you just kind of go at an easy, leisurely pace, you are probably somewhere in the neighborhood of your lactate threshold, but it would take you a long time to accumulate so much lactate that you'd have to stop. But if you go from that leisurely pace to running as fast as you can for half a mile, well, then what you're going to do is because you've increased the intensity of the exercise, and I'll get to the differences in the lines here, but because you've increased the intensity of the exercise, you are going to be using energy, using ATP faster than you can supply oxygen aerobically, and so that lactate is going to start to accumulate. And ultimately, when that lactate reaches a certain point, you're going to have to stop exercising. So there's an absolute maximum point, absolute maximum concentration of lactate before you're going to end up quitting. Couple things. So you can see percent of VO2 max, and again, VO2 max is your aerobic capacity. Aerobic capacity is your ability to supply and use oxygen. So if we have somebody who is untrained, we just have them go out and exercise at half of their aerobic capacity, they're going to start to accumulate lactate. And then eventually they're going to have to stop. If we take somebody who's trained for several months, they're not going to start to accumulate lactate until about 75% of their aerobic capacity. So why is it? How can they run faster before they even start to accumulate lactate? And well, the answer is because of adaptations in the heart. So the heart gets more efficient. So you get a left ventricle that's more extensible, that can contract more strongly. You get a higher blood volume. The trained muscle cells have more capillaries, more mitochondria, more aerobic enzymes. And so because of that, you can use oxygen. You can both supply and use oxygen more effectively. And so if we can use oxygen, well, then we're not going to generate a bunch of lactate, or we're going to be able to get rid of it, is another way of thinking about it. So with training, our lactate threshold shifts and we can work at a higher intensity before we start to accumulate lactates. That's what this particular thing depicts. But what I want you to know, I should finish with that, what I want you to know is lactate threshold is the point at which we start to accumulate lactate or lactic acid. And that's it for this time. I know, like I said, a bunch of pretty complicated concepts, but that's it for Chapter 1. And so we'll kind of revisit these things as we move along, as we talk about specific types of training. But I wanted to get some of the basic adaptation stuff out of the way early on to get you thinking about it. And then we'll talk about types of training that induce these sorts of adaptations. So we will see you next time.